So what I have here is the CRISPR that we prepared earlier. So this contains the gene drive with these regions of homology that's going to be able to integrate itself into the mosquito genome. And so we need to take this DNA and load it into a microinjection needle. In which the end is so small that it can't be seen by the human eye. That's quite careful. You need to be quite sure that there are no bubbles in the end of this, or you can't get enough pressure to inject the eggs. So we need to screw it onto this injection microscope, and it's attached to a source of air pressure over here, which we carefully regulate, and allows us to provide a short burst of pressure that releases a small amount of the DNA that we created upstairs into the mosquito embryos. What we have on this is a number of mosquito embryos that have been placed carefully, all of them tail to head. And we need to inject them right into the posterior of the embryo, because it's this part of the embryo that's going to become the germline, and therefore we can integrate it into the cells that will become the offspring. Now these embryos, they have to be between about half an hour and 1.5 hours old, because if they're too young, the embryos are so delicate that they'll burst when you inject them. If they're too old, they form an eggshell which is so hard that the needle can't penetrate. Now typically, you don't inject one embryo and get a transgenic. In fact, it actually takes many, many hours of practice, and in my case, more than 6 months. So what we tend to find is that we need to inject probably between 100 and 2,000 embryos before you can actually recover a transgenic. So what we do is we wait for these embryos to hatch. Now, very few of them will survive the injection process, typically between 10 and 20%. And out of those, you might expect 1 or 2% contain some parts of the gene drive, but maybe not all of it. So to check that it's gone inside and it's gone inside correctly, we wait for them to hatch and we screen them one by one, looking for the signs of the RFP marker, seeing that they glow fluorescent red. So here you can see the larvae when we just see them naturally under normal light. When we shine the laser through, you can see they look like little light bulbs. And each of these is one of the heads of a larva, and these diamonds down the body are some of the neurons going down. So this red fluorescent protein is expressed specifically in the neurons, which is quite clear in the eyes, the brain, and effectively this is a nerve cord going down the body. And it's very, very easy to identify which mosquitoes are transgenic and which ones are not by looking at this expression.